All right, so on to chapter 3. So we started off, and I mentioned this already in the class. We're starting from the individual atoms, molecules, microscopic stuff, in other words, and then we're slowly building larger and larger and larger. We talked about molecules, macromolecules, the basic stuff we think about probably from a dietary standpoint, lipids, proteins, um, carbohydrates, those kinds of things. And so now we're going to start building those things up, and we're going to look at cells within our bodies. So our bodies are made out of trillions of cells. They're the individual little, as your book uses the analogy, they're the individual bricks when you stack them all together that ultimately make up a building. In our case, it's all the cells that are stacked up together to make us. Your book has a few images just to, again, show you kind of the microscopy involved here. Again, we're dealing with things we can't see with our naked eye. We still need microscopes to be able to see individual cells. So you see all the individual sinus cells here. These are actually plant cells, onion specifically. You can see a few things like the nucleus there and the cell wall. And those are light microscopes, the kinds of microscopes that we use in lab or that you've probably seen typically around for being able to look at stuff. But then we also have scanning electron microscopes that can see things that are even smaller. And so the image on the far right here is an image using a scanning electron microscope. These are images with a light microscope where we can actually see the individual bacteria, and then we can actually zoom in with a scanning electron microscope and see the individual bacteria. They're just dots in these normal light microscope images. But again, if we want to actually see more detail, see things that are even smaller, we can see them actually invading the cell here. You can see a few of them kind of getting down in there. First section of your book that we're not really going to cover here involves microscopes. There's a couple of pages on how cells are studied, and so I would just review those. Of course, it's still fair game for a quiz question in this class, but if you're in lab, Bio 10 lab, then we have a specific lab where we go over how to use microscopes, the typical light microscope and dissecting microscope. Unfortunately, we don't have access to an electron microscope, but in lab we use microscopes a lot throughout the semester, so it's another one of those basic skills as a scientist that you need to have. So in lab we will spend some time going over microscopes and how to use them. But as far as lecture goes, just kind of review those couple of pages there. Some of the things that humans have trouble with are scale, time, those kinds of things. Not so much in the frame of us. So, for example, on this image, move me out of the way here. on this image that is in your book, figure 3.6, it's showing the scale from a human, where obviously we can see humans, and we're in the scale of meters. You start getting into things like eggs, still things we can obviously see with our naked eye. Even frog eggs, even though they're small, they're in that millimeter range now. But if you've ever seen a little cluster of frog eggs in a pond or something, individual eggs are tiny, and you can see the embryo developing in there, but they are visible. even a human egg is just barely visible with the naked eye. Again, now we start getting into micrometer measurements. We start getting into things that are in the scale where they start getting out of being able to see them with our naked eye. But a human egg is right on the very edge of visibility with naked eye. Then we start getting, though, into what we need a microscope for. Obviously, even something we can see with our naked eye, like the frog egg, if we put it under a microscope, it's going to blow it up, and obviously we'll be able to see detail that we can't see with our naked eye. And so then this starts going into what we're going to be looking at in this chapter, which deals with cells. And animal cells and plant cells all contain things that make them work. Our body as a whole contains organs, heart, lungs, pancreas, kidneys. All those organs have a function that help our body work. When you then look at each of the trillions of cells that make up our body, each of those cells inside of them have organelles, which just literally means a small organ. And those organelles each have functions that help the cell survive. So if the cell contains organelles, and each of those organelles are, fulfilling a role, making things that our body needs, then all of those cells are working together to make us function, or to make a plant function. So we'll go over what are in plant and animal cells and how they're different. But again, at that scale, we now need a microscope, a light microscope typically, which again is what we're going to be using in this class if you have the lab. Then we start getting down to the individual little organelles that are inside of a cell. And we can still see those, a lot of them anyway, with a light microscope. But now we're starting also to get down into the range of where we would actually need an electron microscope to be able to see details within those organelles. On one of the previous slides, we saw the individual bacterium that was red in that case. And we definitely need an electron microscope to see the detail of the bacterium. We can see them with a light microscope, just as a little small shape normally. And then when we start getting down to the components of the cell walls and individual proteins and things, then we certainly start getting down into electron microscope territory, because we're looking at things that are in the scale of a nanometer. And so micrometers and nanometers are again, typical measurements that we use as we start getting into things that are too small to see with the naked eye. All right, so as humans started having the ability to see things that were microscopic, because remember, we didn't always know these things even existed. We didn't until we invented microscopes. But there were theories before then, before we could actually see things. There were scientists that theorized that there was more to living things than what we could actually see with our naked eye. And again, as we developed microscopes and so forth, we started developing these what's called a unified cell theory. And basically this consists of the fact that all living things are composed of one or more cells. We'll look at single-celled organisms like bacteria, or multicellular organisms, which could be just 2 cells or mini cells, or billions or trillions of cells like we are. Also, that the cell is the basic unit of life. Remember, there are smaller particles, atoms and so forth, but what makes up something that's actually alive is that it has cells. And again, those cells contain nuclei and different things that we'll look at in this chapter. And the other important part of the unified cell theory is that all new cells only exist or arise, come from an existing cell. So again, just the general idea that a cell doesn't come from nowhere, or a bunch of random stuff doesn't make up a cell. A cell can only come from a pre-existing cell, and that's through the process of cell division, and that's something else that we'll look at in coming chapters. So there are two types of cells, two types of living things, I guess, but two types of cells, prokaryotic and eukaryotic. And just if you're following along in the book, again, my book anyway, we're on page 65, section 3.2, comparing prokaryotic and eukaryotic cells. So pro just refers to as before or maybe first. So it's a before karyotic, which just refers to the way that the nucleus exists or not. So prokaryotic cells don't have a specific nucleus in them, and the nucleus is where the DNA is held. So in a prokaryotic cell, the DNA is just kind of in an area of the cell. It's not within its own little organelle. It's just within a localized area called the nucleoid. And then, of course, it's going to have cell wall around the cell, a membrane. In this case, we're looking at a little maybe bacterium. And so it's got a capsule around the outside. It's got these little pili that are hairs that kind of interact with the environment, a flagellum that allows it to move through the environment, ribosomes, which are the place where proteins are made. And so that's just a generalized view of what a prokaryotic cell might look like. Prokaryotic cells are simpler. They aren't as complex in their structure or their organization compared to a eukaryotic cell. So eukaryotic cells are typically found in more evolved living things. They're a lot more complex. They do have a true nucleus. So any eukaryotic cell that you look at under a microscope, you're going to see a relatively large, circular, darker spot, typically, somewhere in the cell, but maybe near the middle of the cell. Then you may even see a darker spot within that, the nucleolus. And so this is the specific place where the DNA is housed. And then in this lecture, we'll look at all these other things that are labeled here. Things like ribosomes, the organelles like the rough endoplasmic reticulum. There's also a smooth endoplasmic reticulum. An organelle called the Golgi complex or Golgi body, which is kind of like a distribution warehouse for materials that are made in the cell. Lysosomes that go around kind of collecting the garbage in the cell. Mitochondria, which is kind of like where the power is generated. So we'll look at those things in more detail, but it's important to know just the general difference between prokaryotic and eukaryotic cells. And so that's what the previous two slides deal with. So as we go through the next section, we're going to be looking at the different organelles that are in cells and real generally what they do, what those organelles do. Maybe not even everything they do, but a couple of the important things. And then we're also going to be looking at some differences between plant cells and animal cells. And there are some differences in the organelles. that are present in the plant versus the animal cells. And so we'll be looking at a little comparison. This image is actually figure 3.7 on my page 68 anyway. And so this is kind of a good review to go back to maybe prior to the quiz. That'll be upcoming here in the next couple of weeks. And so it just labels everything, basically all of the typical organelles in an animal cell, and generally kind of what they do, and then the same thing for a plant cell and what those organelles do. But again, we'll be going through those as we move through this section. So I'm going to take a quick pause here. I'll try and do this during the lectures. It'll give you kind of a place that makes sense to take a pause if you want to, get a drink or stand up for a minute, stretch, and then we'll come back and pick up talking about the plasma membrane, what the actual membrane of cells contains. All right, so let's get into the actual structure of our cells. So all cells have a membrane that surrounds them, cell membrane, and it's made out of what's called a phospholipid bilayer. Phospholipid meaning the cells are composed of phosphorus and lipids or fats, and that there are two layers of them. So there's one layer that faces the outside of the cell, and each has a polar end. So the polar end is the round head portion there, and it's also hydrophilic, meaning it likes water. And then there's another layer of these phospholipids with the polar end pointing towards the inside of the cell. So the one end is polar, and then the tail end, notice there are kind of like two little tails coming off of each of those. Those are actually non-polar. And so basically that creates this semi-permeable membrane, a membrane that can allow certain molecules anyway to pass through. And then you notice in this simple version here of the phospholipid bilayer that there are other molecules. Some actually go through the membrane completely, like these channels that allow proteins to come and go. There are other molecules like cholesterol that are embedded within the membrane. And then other things, proteins that are embedded in there, proteins that are embedded on the periphery or the edges of the membrane. And we'll get into some of these other things, glycolipids, lipid and carbohydrate molecules that are embedded in there, glycoproteins, proteins and carbohydrate molecules. And so this phospholipid bilayer then basically forms the protective layer around every cell that protects what the inside contents are. And so again, the key thing there is that it's a phospholipid bilayer, two layers of phospholipid molecules. We'll actually look at a little more detail of this in just a bit. So some of the other components within our cells are a variety of filaments, microfilaments, just meaning microscopic little filaments or fibers. There are intermediate filaments as well, microtubules, and basically these form a few different functions. One of the main functions is they form what's called a cytoskeleton, the skeleton within the cytoplasm of a cell. So again, here's our eukaryotic cell, and we'll talk about these organelles, again, the nucleus. But the rest of the space inside the cell membrane that we were just talking about is filled with cytoplasm. And if you look at the cytoplasm under a microscope, you'll see a variety of things like these filaments that, again, basically serve the function of being a skeleton. The other place we see the microfilaments and the microtubules come into play is when we're going to talk about cellular reproduction. Those microfilaments and microtubules help basically a cell divide into two copies of itself. So we'll save that for later. But again, within the cytoplasm, you have these filaments that basically form a structure that keep the cell rigid, basically, although not really rigid so much as just it kind of holds it together, it gives it support structure. All right, so if we zoom in then to some of those organelles that are inside the cytoplasm, one of the really important ones is called the endoplasmic reticulum, and we actually typically abbreviate that ER for endoplasmic reticulum. There's actually a rough endoplasmic reticulum and a smooth endoplasmic reticulum. So RER and SER. So this image, which is on page 71, figure 3.10, if your book is numbered differently, shows the nucleus. So this is zooming into the nucleus, and then the nucleolus. And remember, the nucleus is where the DNA is housed. Immediately around the nucleus is what's called a nuclear envelope, which is just kind of again, kind of an outer protective covering. And then in close proximity to that is the endoplasmic reticulum. And so again, the nucleus is where the DNA is housed, and it's in the form of what we would call chromatin. The DNA goes through a variety of processes when the cell reproduces, but again, we'll save that for a little later. The nucleus is also kind of the brain of the cell, and so it actually directs the synthesis of the ribosomes, which are represented on this figure by all the little red dots, as well as the proteins that are synthesized inside of those ribosomes. And so again, our body, just in the big picture, right, takes in raw materials, the food we eat, in other words, and then it's broken down into the basic components, and then the organelles like the endoplasmic reticulum and the ribosomes more specifically collect all those raw materials from what our body takes in, and then it recreates the specific molecules that it needs, and then they're shipped off to wherever they're needed in our body. And so again, we'll talk more about the ribosomes and protein synthesis in just a few minutes. But as you look at this nuclear envelope, notice there are spaces all around the nucleus. These are pores, or basically just passageways or openings to get from outside the nucleus to the inside of the nucleus. And remember also that the boundary around the nucleus is just like pretty much any other cell membrane, in that it also is a phospholipid bilayer. The only difference in this case is there's actually two bilayers, two layers that each have two phospholipid molecules arranged, head outside, tail inside. And so these pores, like any other opening, allow for exchanging things. Could be ions, various molecules, the ribonucleic acid, which is also important for making new copies of DNA, for example. So here now we zoom in a little more. Again, here's the nucleus for reference. You see the rough endoplasmic reticulum. So this is figure 3.13 in your book. Again, this is the rough ER, and it's called a rough endoplasmic reticulum because with all of these ribosomes on the surface, it makes the surface look bumpy or rough, and so that's why it's the rough ER. And so this is composed of what's called the endomembrane system, and this endo inside the membrane, so the system that's inside of the plasma membrane out here, basically works as, you think of, for example, you go to a store like Target, and they've got all this stuff in there that you can buy. Well, that stuff had to come from somewhere, so from like a distribution plant, and then even the distribution plant then had to get the individual raw materials or the individual things that it was repackaging and shipping out to all of its individual stores. But first, of course, everything that you buy has to be made somewhere. And so that's kind of what's going on here. So the rough endoplasmic reticulum, and specifically the the ribosomes on the rough ER, actually make proteins. So again, they're getting all the raw materials from what we eat. They're making the proteins. They're also making the phospholipids, which obviously we already talked about. If all of our cell membranes have phospholipids, then to make new cells, you need more phospholipids. And so what happens then is these proteins, and so again, here's a little protein. It ends up getting synthesized within these ribosomes. Then it gets packaged, and the package, which you could think of as being like a semi or a train, right? Semis back up to a factory, they pick up the stuff, and then the semi drives it to, say, again, a distribution center before it goes out to the individual stores. So these are transport vesicles, which again are kind of like a semi-truck that carries stuff from somewhere to somewhere else. And so this vesicle actually forms on the outside of the rough endoplasmic reticulum, and it basically forms a little protective bubble around the protein. That protein then is transported, and it's only a short little distance, but it's transported from the ER to the Golgi apparatus, which is basically kind of like the distribution center. So the factory builds something, it gets shipped, in other words, through the transport vesicle to a distribution center, and then the distribution center, or in this case again it's called the Golgi apparatus, actually kind of resorts and repackages the proteins and basically gives them instructions on where they're needed inside of our body. So the transport vesicle connects to what's called the cis face of the Golgi apparatus. So there's kind of an inside and an outside to the Golgi apparatus. And so the vesicle attaches to the inside, it releases the protein. The protein gets, again, repackaged, gets instructions where to go. And then another transport vesicle picks the protein up on the other side and then transports it outside of the plasma membrane, outside of the cell membrane. Notice the vesicle kind of bumps up against the membrane and then opens up and allows the protein to go out into the bloodstream or get transported basically wherever it's needed in the body. So again, the endoplasmic reticulum makes the stuff like proteins and phospholipids. They get transported to the Golgi apparatus, which again Again, basically gives it instructions where to go, does any repackaging necessary, sends it off, and then it goes out to wherever it's needed in the body. Remember, proteins are really important in muscle, for example, a lot of other things, but building muscle, one of the needs for protein. And then we also have the smooth endoplasmic reticulum. Smooth endoplasmic reticulum also builds things, or what we would call synthesize. And so the rough ER synthesizes proteins and phospholipids primarily, whereas the smooth ER synthesizes carbohydrates, lipids, steroids, which are a type of lipid. But also a really important role is at this individual cell level. The smooth ER also helps take toxins out of your body. And that could be medications you take, because remember, those are foreign to your body. Any sort of poisons or anything the body perceives as a poison. And that would include things like alcohol, for example. So the organ in your body that is responsible for a lot of the detoxification is the liver. I was kind of going slow to give you a chance to think about it. But then at the individual cellular level, the microscopic level, the smooth endoplasmic reticulum is the specific little organelle that is responsible for the detoxification process. So three very important organelles, the rough ER, the smooth ER, and then all of the stuff that's created there gets sent to the Golgi apparatus, where it gets repackaged and kind of told where to go. So as we're going through these different organelles, and for quiz purposes, just make sure you understand what the organelles are actually doing, what their primary purpose is. Again, the questions on the quizzes for this class will be multiple choice, or maybe some matching, that kind of stuff, but you'll have options to choose from. So those were obviously drawings, idealized drawings. Here's an actual image. A micrograph is what we would call these. Notice anytime there's a figure, or in this case, an image of a cell, they always have the scale put in there so you can tell what the scale of it is. So again, this is actually, I think this is figure 3.11 in your book. So this is an electron microscope image, because again, we're talking about things that are too small to see with a typical light microscope. And so you can actually see the Golgi apparatus here. Remember on the previous slide, it's got these folds in it, which are called cisternae. And so that's what's represented here. See all the different folds, and the cis face side of the Golgi where stuff goes in, and the trans face where the materials are taken out. So here's just more of a close-up of the Golgi apparatus to see what's happening. Again, a transport vesicle goes to the receiving side of the Golgi. It opens up, it releases the proteins, phospholipids in this case, from the rough ER. They go through this kind of maze-like process. Again, they get resorted, reorganized. They're given instructions on where they need to go. And then when it's ready, basically, the material gathers up, and you have a new vesicle forming. You can see all these vesicles. Anywhere there's a bit of a bulge, there's a new vesicle being packed with stuff that needs to be transported. And when it's full and ready to go, then it just kind of pinches off, and then it goes out to wherever it's needed in the body again. So a couple of really important organelles. another really important, I guess they're all really important because we need them. They wouldn't be there if our bodies didn't need them. But lysosomes are the next thing we're going to look at. It's always kind of easy to use these catchphrases to maybe help you remember what the purpose of things are. So the lysosomes are kind of the garbage disposal. The nucleus is the brain. It's where our DNA is held. The rough and smooth ER are our factories, protein, phospholipid factories for the rough ER, carbs, lipids, etc. for the smooth ER. The Golgi is kind of the distribution center. And so lysosomes are the garbage disposal. Anytime we have something that's in our body that isn't supposed to be there, say for example, some sort of pathogen, you get a bacterium in your body that isn't supposed to be there. The way that our body deals with foreign objects is the chemical signals are there that something is there that shouldn't be there, and then basically things like lysosomes are dispatched. Within the macrophage, like a white blood cell, for example, we have these lysosomes. So it's the macrophage's job to actually hunt down and find in this case, a bacterium. And so then it engulfs the bacterium, surrounds it by a process called phagocytosis. Phagocytized is what it's called when in this case, the white blood cell surrounds the offending molecule there. Once it's kind of safe inside of the white blood cell in this case, it's packaged off in a vesicle. So vesicles are typically, used for surrounding and transporting other things. Same in this case. And then what happens is the lysosome, which is inside of the white blood cell, finds this foreign object, merges with it, and then it releases enzymes, hydrolytic enzymes, basically that can break up and destroy the bacterium in this case. So again, the white blood cell itself actually is roaming around, and that's why, for example, your doctor might draw some blood if you're feeling sick, and they do a white blood count. If you have an infection of some sort, your body starts producing more white blood cells, and so there'll be more of them in your body. And that's a bit of indirect evidence to your doctor that you probably have an infection. So your body generates more of these to hunt down whatever the problem is, and then the lysosome inside of the macrophage breaks it down, renders it harmless, and then whatever's left over can actually leave the white blood cell and be disposed of completely outside of the body. So lysosomes within macrophages. If the nucleus is sort of the brain, then the mitochondria would be considered sort of the powerhouse or the generator of our cells. This is where the primary energy carrying molecule, which is called ATP, short for adenosine triphosphate, that's where ATP is made. So your body can't make proteins or carbohydrates or make more white blood cells, make lysosomes without energy, right? Nothing is free. So for your body to do what it does, it needs to make energy, and the primary type of energy that our body uses at the cellular level is called ATP. Muscles, for example, do a lot of work, so they have a lot of mitochondria. If you have tissues in your body that don't work as hard, that don't have as difficult a job, then they might have a lower concentration of ATP. But again, places your large muscles, especially in your legs and arms, your heart muscle, etc., because it needs to do a lot of work. It's always moving. It needs a lot of energy, and so there are going to be more mitochondria in the cells within those specific areas of your body. So this figure is 3.14 in your book, and basically it just shows the membrane of the mitochondria, which you can kind of trace that around. And then there's other parts inside of here, but fortunately we're not going to get into how the mitochondria actually works. Your body needs them obviously to make energy. Your book has a paragraph, a little bit of description for each of these organelles. So read through that. It talks basically about how the mitochondria generates ATP, but again, we're just trying to give you an overview to make sure you at least have a basic understanding of how our bodies work. So then on my page 74, it looks like, there's a section that's called animal cells versus plant cells. And so it talks about some of the things that are different between animal cells and between plant cells. A couple main things is animal cells. We have organelles or structures called centrioles and centrosomes. And those are both involved in cell reproduction. So how an animal cell reproduces is mostly the same as how a plant cell reproduces. But at the very end, when you go from one cell and then you end up with two cells, how those cells actually separate from each other is involved in again, why animal cells have these structures, whereas plant cells actually form a hard cell wall. And again, we'll talk about cell reproduction later. We also just talked about lysosomes. Plant cells don't have those little things inside of the macrophages that actually are released to break down foreign objects. Plant cells have a different way of dealing with that. And then a couple of the main differences with plant cells is instead of having a membrane, a cell membrane, phospholipid bilayer, they actually have a more rigid cell wall. Plants make their own energy. So whereas the cells in our body have to actually depend on our body taking in food, and then the cells convert that energy from one type to another type, but our bodies can't make their own energy. We have to eat food. Whereas the main difference with a plant is that plants do make their own energy within the chloroplast. Plants are able to convert sunlight into simple sugars, glucose, and then that's the kind of basis for our diets. So plants basically have chloroplasts which allow them to convert sunlight into other types of energy that we then depend on. There are also a couple of ways that plant cells are connected to each other. We'll talk about the plasmodesmata, plastids. And also another obvious difference is that plant cells have what's called a central vacuole, which is kind of a big empty space, which can be used for storage. So whereas our cells don't have kind of storage devices inside of them, plant cells do have these storage organelles called vacuoles. So in another chapter, we'll talk about the process of photosynthesis in a little bit of detail, not as much as what you'd see in a biology majors course, but still a little more detail. For now, though, just kind of the structure of a chloroplast. So it's got an outer membrane and an inner membrane, so kind of two layers. Inside of that second membrane are these stacks of what are called thylakoids. Think of like a bunch of plates stacked up in a counter in your kitchen. Each one of these is individually a thylakoid. The entire stack of these thylakoids together is called a granum. And then in between those granum is just space, sort of liquid space. And that space is actually called stroma. So in an animal cell, that empty space, that intracellular space between the cells or between the organelles, or the space inside of a cell between all the individual little organelles, is cytoplasm. In a plant cell, though, when we get inside of the chloroplast, then we have this liquid called stroma. So again, just the basic structure of a chloroplast, which is where photosynthesis happens inside of plants. So again, we have billions to trillions of cells in our body, and in the space between those cells, there's a lot of other things going on. So again, to put it in perspective, here is a cell membrane, and this side would be outside of the cell, which we refer to as extracellular, outside or beyond the cell. The bottom part here would be inside the cell, intracellular. So outside of the cell, between cells, are going to be a lot of different things like collagen fibers that, again, just kind of give structure and support. You can kind of zoom in on these different fibers here. You can see they're made out of carbohydrates, proteins, polysaccharides, which are long chains of sugars. So there's this kind of microscopic network of these fibers that basically allow the spaces. So here's a cell. In between that would be all of those collagen fibers, and it basically allows the movement, so cells can kind of move and stretch from each other, but not totally fall apart. So these collagen fibers, fibronectin, all of these things work together to basically give the cell movement, or the space between cells movement, but not to where they break or anything. And that was figure 3.15, or is that 3.16? 3.16. The next thing is that each cell has to communicate with its neighbors. And communication in this case might just mean moving stuff from one cell to the next. And so there are different ways that cells can communicate with each other. There are types of connections called plasmodesma. And so these are channels between cell walls of two adjacent plant cells. So again, these would be the way that two plant cells are connected. And so here's one plant cell, here's the other one. And there's basically just this little opening, this little channel called, again, a plasmodesma, which allows materials that are circulating inside of this cell to be able to go into that cell, and then the next one and the next one, until that material gets wherever it's needed inside of the plant. In animal cells, there are junctions, a couple different kinds. Tight junctions help keep cells from leaking. So again, remember, there have to be openings in cells, channels, that allow stuff to go from one cell to another. If those channels were open all the time, and there wasn't anything kind of regulating stuff going in and out, then the cells could just leak all their contents out into the extracellular space, and that wouldn't be good. So these tight junctions, so here's one cell wall here, and the cell goes off this direction. Here's another cell, and it would continue going off in this direction. This is only showing one little sliver of the cell, but keep in mind it keeps, the cell would be out here. And so where the cells actually touch each other are these tight junctions. So these almost like weld spots in a way, although the desmosomes are also kind of referred to as welds. But basically you have these little openings, so stuff that's in this cell can go through any one of these openings and get into the next cell. But again, that basically allows materials to go from one cell to another, but not to just leak outside of the cell somewhere. Here are two cell walls again, one here, one there. They're also called desmosomes, which are like spot welds. So they allow the cells to move, to stretch, but not tear apart. And then also things that are called gap junctions. And these actually have the ability to open and close. So again, you wouldn't always want there to be an opening between two cells. You don't always want stuff just to move through. So when the material needs to stay in place, the little gap junction stays closed. Kind of looks almost like a little flower, right? It's called a connexin. And so it stays shut, and then when material needs to pass through, it'll open up and allow material to go from one cell to another. On my page, 78 and 79, and I think on to page 90 in my book, figure or table 3.1 lists each of the components of the cell, the function of those components, whether it's present in prokaryotic cells, and then whether it's in an animal cell or a plant cell. And remember, a lot of the organelles are going to be present in both plant and animal cells. Some things are going to be both in eukaryotic and prokaryotic cells. So again, just a good maybe a place in your book to put a little sticky note or something, so that if you're doing the quiz and a question comes up about the organelles, then if you don't remember the answer to the question, you can just go to that spot in the book and find your answer. All right, so just a little more about the cell membrane now. Again, we got the same image of the cell membrane that we looked at earlier. As we've, again, started getting the technology to look at these things that are microscopic, we developed what's called a fluid mosaic model that we use to describe basically this plasma membrane. So it's not really a solid like a wall of your house. Your house has openings, right? Doors. In other words, things like these protein channels that allow you to get from the outside of your house to the inside of your house. And the walls of your house has stuff like insulation and blah, 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 blah. But we think of that as being a solid object. You can't just walk through the wall. What's different about a cell membrane is it's kind of a little more like a fluid than a solid. So it's not just this rigid structure. Again, yes, there are these channels, and so larger molecules can only get through your cell membrane by going through one of these channels. But smaller molecules, water molecules typically, but a lot of other small molecules are actually small enough they can squeeze through, and they can kind of come and go depending on some other things that we'll talk about here in just a few minutes. So again, don't think of the cell membrane as being just like a balloon or a plastic bag or something. It's really more of a liquid kind of material, because again, some things can just pass right through them. So again, phospholipids make up the two layers here, the head and the tail part facing out from each other, a phospholipid bilayer. Again, our body does need cholesterol to function properly. Too much is bad, but our body does need cholesterol. And then, of course, there are proteins and carbohydrates all embedded in there, and they all have a variety of functions or roles that they play. And again, there's a section in your book that talks about the fluid mosaic model. It goes back to being described in 1972 by a couple of scientists, Singer and Nicholson. And so it talks again about each of the components and what they do. I guess the one interesting thing I'll mention, and it is in your book, is cholesterol. And again, when we think of cholesterol, it usually comes up in a bad connotation, right? Got high cholesterol, which could lead to heart disease and hardening of the arteries. But in our cell walls, it actually serves as kind of like an antifreeze. It keeps the cell membrane from getting too solid. And so it plays a really important role. There are, you think of any animal that lives in a cold environment, polar bears, penguins, they're going to have a lot more cholesterol in their cell walls so that their cells can resist, getting too cold and then kind of freezing up. So you tend to find that animals that live in cold environments tend to have a higher concentration of cholesterol within this cell wall. Frogs hibernate in the winter time, right? And if they live, say, up in Wisconsin, where I'm originally from, and where the lakes freeze and it gets cold for 3, 4, 5 months out of the year, frogs will typically just kind of settle down to the bottom of a lake, kind of work their way into the mud when it gets cold, and then their bodies just go into a hibernation mode. The water can freeze, and yet the frog can still thaw out. You maybe even have seen this on TV where they have a frog that's literally been frozen solid. You tap on it, sounds like a piece of glass or whatever, rock. And yet when the temperature warms up and the frog thaws out, it can come back to life. We can't just freeze a human and thaw them out and they're fine. If cells don't have enough cholesterol in them, they freeze. And remember in the previous section, we talked about properties of water. The fact that property of water when it freezes is that it forms that lattice, right? And it expands. Again, think of a cell in your body that freezes, and because there's a lot of liquid in there, it expands, and when it expands, it can actually rupture the cell. And when the cells all break in your body, there's no way just to fix them. So again, frogs can be frozen basically because they have a lot of cholesterol or antifreeze in their cell walls, which allows them to basically resist their cells popping when they freeze. So again, there's a section about the fluid mosaic model. All right, so this would also be a good point to take a brief pause then before we get into the last section of the book that deals with movement of molecules, transport of molecules from one place to another. And we'll run through some terminology that addresses how and why molecules move from one place to another. All right, so stuff that's made in our body, again, the proteins, carbs, whatever, liquids in general, need to be able to move from one place to another. They can move by being held in those vesicles and then just being transported to where they're needed. But a lot of time, the substances that have to kind of come and go, leave and enter our cells, go by a variety of different ways. One of the main ways is just passive transport. Passive transport doesn't require any energy. So we compare that to what we're going to look at in a minute, called active transport, where you actually have to take energy. So passive transport, again, imagine you've got a big rock on the edge of a hill. You can just kind of give the rock a little nudge, and because it's heavy and because there's a slope, it's just going to go downhill on its own, basically. You don't have to keep pushing it downhill. So that's kind of what passive transport would be. Basically, substances moving from one place or one cell to another, kind of in a downhill fashion. And we'll talk about what downhill means in a cell compared to active transport, which would require energy. You can't just push a rock, give a rock a little nudge uphill and expect it just to kind of keep going on its own. You have to keep pushing it to get it to go. Our cell membranes are what's called selectively permeable, or they have what's called selective permeability. If anything could just come and go as it wished, our cells wouldn't be able to maintain themselves. Because again, you don't want things just kind of coming and going without any kind of control. Again, imagine, your doors are unlocked in your house and people just kind of came and went as they wanted to. Somebody comes in, grabs the pizza out of your fridge and heads out. We've got doors that basically limit the ability of people to come and go from our homes. And the cells, as we saw, have those channels and the phospholipid bilayer that makes it selectively permeable. So the proteins that are in the cell wall that we saw on the previous images work as channels, and things can be pumped in one direction as they're needed. You have things like lipids that can easily go through the phospholipid bilayer. Again, smaller molecules can come and go. Oxygen, carbon dioxide can go straight through the cell membrane by a process called diffusion. And that's going to be the next thing we look at. So just remember, our cell membranes have selective permeability. Not just anything can pass through our cells. So the next process we're going to look at, and this is basically one of those passive types of transport, is called diffusion. So again, notice we have a membrane here, right? A phospholipid bilayer, so a typical plasma membrane. And notice that we're going from left to right, time, just minutes, whatever the time frame is. So this is the outside of the cell, and this side is the inside of the cell. So when you've got fluid on the outside of the cell, as long as the molecules are small enough, they're going to move from along what's called the concentration gradient. So notice on this side of the cell, there's a lot higher density of this fluid, whatever this fluid is. And there's a lot less of it on the inside of the cell. So because of the difference in concentration between the outside and the inside, diffusion is going to happen. Again, as long as it's going through a semi-permeable membrane along a concentration gradient from areas of high concentration to areas of low concentration. And ultimately, what's going on here is that we're seeking balance. So if there's a higher concentration there and you have a semi-permeable membrane, these molecules are going to diffuse through the membrane from the outside to the inside over time until the concentrations are equalized on the outside and the inside. Again, this is a passive process. No energy is required. It just happens because there's a difference in the concentration gradient. So that would be a type of, again, a type of passive transport called diffusion. The rate of diffusion, or how quickly it happens, is affected by a few different things. How big of a difference there is in the gradient. So for example, going back to this slide, if there's a lot of something on this side and nothing on the other side, in other words, there's a huge difference in the concentrations on both sides of the membrane, the rate of diffusion will be much quicker than if the concentrations are more similar. So how big of a gradient there is, in other words, how much of a difference there is in the concentrations on the two sides of the membrane, the greater the gradient, the faster diffusion is going to occur. The size of the molecules, bigger molecules, the more massive molecules are going to diffuse much more slowly. Again, just imagine you've got a dozen 200-pound perfectly round rocks in your lawn, and you wanted to kind of roll them around your lawn to space them out. It's just physically going to take you a lot longer to roll 200-pound rocks than if you've got a dozen 2-pound rocks. You can pick up the 2-pounders and even just kind of throw them around the yard. So you can make the small molecules move much quicker. The larger the mass of the molecule, the slower the diffusion. How do you think temperature affects the rate of diffusion? If this water was cold, let's say, close to freezing, and then we had another container of water that was 180 degrees, and you put a drop of food coloring in those two, a cold water and a hot water, which one do you think would have a quicker rate of diffusion? Hopefully you guessed the hot water would have a quicker rate. Molecules, when they're heated, they start bouncing around, right? That's what causes water to boil. The molecules get warmer, they start bouncing around, they're bouncing off of each other, and eventually when there's enough heat applied and those molecules really get energetic, you get boiling water. Well, again, if the water is warmer, that means the molecules are moving around, the water molecules, and so you put a drop of food coloring in there, just the movement of the water molecules is going to cause diffusion to happen much quicker compared to if it was in a cold water container. And then also the density of the solvent. If you had water and then you also had a same size container of oil, oil is much denser than water. So again, you put a drop of food coloring in the water, a drop of food coloring in a container of oil. It's going to take a lot longer for that food coloring to diffuse through the oil because it's a lot more dense. So that's diffusion. Again, it's movement of water from a concentration from a lower concentration to an area of higher concentration. And so that's how water might move through our body, through our cells, just by simple diffusion. But sometimes the membrane is a little more semi-permeable. It may even be the membrane may even have such small spaces between the cells that water can move through it, because water is a relatively small water molecule. But you might have other molecules in there that are much larger, and they're too big to kind of go through that membrane. So they would be too large to pass through that phospholipid bilayer. So again, things always want to be equalized. They always want to come to a nice balance. And we're actually going to do an experiment in lab, if you have lab, that demonstrates this process called osmosis. So same idea is happening here, but in this case, the molecule here can't move through the membrane to equalize itself like it did in this figure, right? These molecules were small enough they could go through the membrane until they were equalized. In this case, these molecules are much too large to go through this semi-permeable membrane. But yet again, things want to come to a balance. So because these molecules can't go through and equalize, what happens is the water passes through the semi-permeable membrane until basically the density is the same on both sides. So notice in this case, there was a higher density of this molecule on the right side of the membrane. And because these molecules couldn't go to the left to balance out, the water went in the direction of the higher concentration, and the water kept passing through the membrane via osmosis until the density is the same on both sides. Now notice again, there's more water on the right side, but there's also a lot higher density of these molecules. Less water, but fewer of these molecules. But so as far as density goes, the amount of space that each of these molecules has within the water is now the same on both sides. So because in this case the molecule couldn't diffuse through the membrane to equalize the density, the water had to diffuse through the membrane via osmosis to equalize the density. So again, this process is called osmosis, when the water moves through the membrane to balance out the density, as opposed to when the molecule is able to move through the membrane to balance out the density. So diffusion and osmosis, make sure you understand the difference between those terms. Next thing we're going to look at is called tonicity. And this has to do with what we were just talking about, the relative concentrations of solutes in a solution. So going back to this image, these little purple molecules would be the solute, and then this pinky colored liquid would be the solution. It's like you add salt to boiling water for your pasta. The salt would be the solute, the water would be the solution. So tonicity refers to the relative concentrations of solutes in a solution. So there are three terms that we can look at that refer to the possibilities in relation to the tonicity of a cell. And these are hypertonic, isotonic, and hypotonic. So tonic refers to tonicity, the concentration. Hyper means greater, right? In Reading in the summer, you have to be careful so that you don't, get heat stroke. And that would, because your body's temperature raises to a higher point. And, excuse me, I got distracted here by something happening in the room. So we use the example here of blood cells. So these are all red blood cells that are in a solution, and we would refer to them as being hypertonic because the water is leaving the cell to balance out the concentration of solutes. So there's a greater concentration of solutes outside of the blood cell. So there's a greater concentration of solutes outside the blood cell. So imagine the left of this membrane is inside of the blood cell, and this is the outside of the blood cell. So then what's going to happen is the liquid inside of the blood cell is going to want to leave the blood cell to try and balance out the concentration of whatever solute is there. And so again, as you can see from this example, that means the water is leaving the blood cell. And by the water leaving the blood cell, you're dehydrating the blood cell basically, which isn't good. The exact opposite of that would be the hypotonic solution. So in this case, the exact opposite is the case. The concentration of solutes is much lower on the outside of the blood cell. So the stuff, the solutes that are inside the blood cell are much greater than the concentration outside of the blood cell. So now you have water going into the blood cell to try and dilute what's in there. And that can not only cause the blood cell to swell, but if water keeps going in there, eventually the blood cell is going to burst because it won't be able to contain all of that water that's going inside of it. So that would be a hypotonic, and again, we're referring to the solution here. So in this case, the solution is outside of the blood cell. You've got the inside of the blood cell and then the solution that's outside. So if outside of the cell has a greater concentration of solutes, we call that a hypertonic solution. And again, that results in water leaving the cell, which causes it to shrivel up. If the concentration of solutes is much higher inside the cell than outside of the cell, it's a hypotonic solution. And so you have water going inside the cell, which can burst the cell. But in the way it should be, in a balanced situation, you would have an isotonic solution. Iso meaning similar or same. You're going to have about the same concentration of solutes outside as inside the cell. So you're still going to have water moving inside the cell, but then that's going to be balanced by water moving out of the cell because the concentration of solutes is similar enough. So this can get confusing quickly because you can, if I'm referring to the solution, then it's the solution that has either a hypertonic condition, more solutes outside, in other words, more solutes in the solution than inside the cell. Hypotonic, meaning there are less solutes in the solution than inside the cell. Or obviously the easy one is iso. Sometimes when you hear or read talk about tonicity, it might say something like a hypertonic cell instead of a hypertonic solution. So remember, if it's saying cell, then it's talking about where the concentration is. A hypertonic cell would have a higher concentration of solutes in the cell. Now notice again, The one I'm circling here is a hypotonic solution. But then you could say it also are hypertonic cells. It's basically the exact same thing. You just have to specify whether it's the solution or whether it's the cell. So this can be both a hypotonic solution and a hypertonic cell. This can be both a hypertonic solution and a hypotonic cell. Okay, so something that you probably need to go back and think about and read a little more about, because this is really, trust me, I've been teaching this a long time. This is an area where it's easy to get confused. So make sure, again, first you know whether you're talking about the solution or the cell. And again, a solution that's hypertonic would also then mean that the cell is hypotonic. All right, so again, that can go both ways. So just work on that one in your own head, and by reading it and thinking about it, maybe drawing some sketches to help remind you, because there will be quiz questions or test questions that deal with tonicity. Exact same thing with a plant cell. So again, we were looking at animal cells, specifically blood cells. Plant cells have a lot more of a rigid cell wall, right? They don't have a cell membrane, they've got a cell wall. But the exact same thing can happen. A hypertonic solution, again, means that there's more solutes outside of the cell, which means the water is going to leave the plant cell to try and dilute what's on the outside. So again, this would also be a hypotonic cell in a hypertonic solution. So here are just, again, examples of hyper, iso, and hypotonic, showing you the direction of the water movement. Now, plants actually like a little bit more pressure. They like to have a little bit of a hypotonic state, which creates what's called turgor pressure, right? So if you think about there being a little bit of inflow of water, notice the cell is a little bit bulged compared to one that's isotonic, right? So there's a little bit of pressure on that plant cell, and that's actually what helps keep plants upright. I've got a couple of herbs in my garden that need water every day. And if you don't water them when you should, you wait a little too long, they start wilting, right? We've all seen a plant that's wilted. That's because what's happening is the water is leaving the cell faster than it's being replaced, because there isn't enough water in the ground. You didn't water it recently, so the cell doesn't have the ability to keep water kind of pushing inward. And so then when it loses its turgor pressure and the water is leaving the cell, creating a hypotonic cell, then the pressure that helps support that cell wall allows the plant to droop or wilt. So plants that are adequately watered maintain, every cell in that plant maintains a little bit of pressure, and that allows the plant to be upright and rigid. All right, so the last concept, the last type of transport that we need to talk about deals with electrochemical gradients. So again, we have a cell wall here. We have inside of the cell is on the right. The outside of the cell is on the left. And again, you should be able to identify that, because we know that cells have cytoplasm inside of them. And then again, this is labeled extracellular, outside of the cell. So there are these electrochemical gradients. So it's partly based on positive and negative charges, but it's also based on chemicals and them being balanced. And so these gradients, and a gradient's just a difference, right? A gradient is basically just a difference in positive and negative charge, a difference in chemicals or molecules. And so between there being different concentrations of things, notice there's a lot higher concentration of potassium inside of a cell than there is outside of the cell. So there's a concentration gradient here because there's a difference, higher concentration of sodium or potassium versus a lower concentration. Also, same thing with sodium. There's a concentration gradient of sodium because there's a lot more of it on the outside of the cell than there is on the inside of the cell. So you've got inside of the cell, you've got proteins, lots of protein molecules. And so they're not shown here. Potassium isn't protein. We're just indicating that there's a lot of protein inside of these molecules that our bodies need for a lot of different things. Proteins carry a slightly negative charge to them. So if there are lots and lots of proteins, there's going to be a lot of negative charges, which generally results in a negative charge inside of cells. So every one of our cells has a slightly negative charge to it. Whereas on the outside, because of the molecules out here and the fact there aren't all these negative charges from proteins, on the outside of the cell you have a slightly positive charge. So that creates an electrical gradient. Positive charge versus negative charge. That difference creates a gradient, as well as the concentration gradient that I was talking about with the higher concentration of potassium on the inside, higher concentration of sodium on the outside. So your body is going to tend to try to balance itself out. So these potassium molecules are going to want to kind of leave through these protein channels to try and balance out the concentration. The sodium molecules are going to want to kind of find their way inside to balance out their concentration. But the problem with that is that our body needs to have all of this potassium on the inside, and we need the sodium not to be inside the cell. So our body's constantly trying to work against the normal process of these molecules trying to reach an equilibrium. So our bodies need energy basically to be able to keep the balance. Now again, it's not a balance that finds itself easily, right? Again, these molecules want to leave to find a balance, but our bodies don't want them to leave. So we have to keep pushing them back in against the concentration gradient. So, and this is on, section 3.6 in your book, page 87, starts talking about electrochemical gradients. So, the way our body deals with those imbalances, trying to maintain those imbalances, is through this process of sodium-potassium pumps. So again, we were looking at the previous slide, right? We've got the sodium out here, the potassium in there, and potassium wants to go that way to equalize, sodium wants to go that way to equalize, but our bodies need there to be more potassium on the inside, and it needs to have the sodium out there. So the way that works is through this kind of complex process. This is basically the same, it represents the same thing that's in the figure 3.24, right? It's just representing it in a little different way, and then we're going to see how these pumps work. I think, if I remember right too, if you look at the actual PowerPoint lecture, you can click on this link here, and I'm pretty sure it takes you to a nice little graphic, little visual aid, which also brings up a good point that any of these topics, a lot of them we're gonna talk about in lab, so you'll get a chance to do some hands-on experiments that'll demonstrate diffusion, osmosis. But otherwise, I would really recommend going into YouTube and searching for some of these things, because there are lots of great animations on there that will help you visually see what's happening, not just this static image, but an actual video. And so I do have links to some of those as you go through the lectures. But otherwise, again, just Google sodium potassium pump and you'll find all kinds of great animations that'll help you understand it. So again, we've got the outside of the cell on the top, the inside of the cell on the bottom. We've got all of that potassium. Again, higher concentration of potassium, but a lower concentration of sodium inside the cell. And then the opposite is true on the outside of the cell. But again, because these, excuse me, losing my voice, a good thing because we're getting close to the end. These potassiums are just diffusing out of the cell to try and balance out their concentration gradient. So they're getting to the outside of the cell. And what we have to do then is actually pump them back in the cell, force them against the concentration gradient back inside the cell. So on the far right here depicts the concentration gradient. Potassium, much higher concentration on the inside than the outside. Sodium, a much higher concentration on the outside than the inside. So these potassium, are leaving the cell, but yet then they have to be forced back in the cell. So kind of like pushing a rock uphill. It doesn't just roll uphill by itself. It rolls downhill easy, but then we have to get it back to the top of the hill, which requires energy. And again, remember that primary type of energy at the cellular level is ATP. So we get these sodium potassium pumps where basically the potassium then is gathered up in these channels, and then the ATP, little energy packet, is actually used to push the rock up the hill. Again, it takes energy to do that, right? And it takes energy to push something against its gradient. So these ATP molecules, notice they're adenosine triphosphate. So these are 3 phosphate molecules. And the phosphate breaks off in the process. It releases energy to be able to do this work, which then creates an ADP, adenosine diphosphate molecule. And so then it forces those potassiums back inside, and it gives off this 1-phosphate. And then basically the same thing is happening in the other direction, right? The sodium is kind of diffusing inside, again, to try and balance out the concentration. But again, we don't want all that extra sodium on the inside of our cell, so it has to be forced back out. So again, those sodium molecules get collected in the channels in the pump, and then with ATP forces those sodiums again against their gradient back on the outside. So there's this constant use of energy just to maintain the balance inside of our cell. Again, a lot of things can just kind of come and go, and they're going to do that to balance out. But because our cells don't function if there's too much potassium or not enough sodium, we have to actively use ATP to force these molecules against the gradient so that we can maintain that proper balance. So, so that figure that we were just looking at is figure 3.25 on my page 88, I guess. And so again, it talks about how these pumps work to move these molecules against their concentration gradients to, again, maintain balance in our bodies. All right, then the last couple of terms. Our process is called endocytosis. These involve vesicles again. Remember, vesicles are just little devices, little things that surround something and basically are like a semi-truck. They contain something and then ship it off somewhere else. There are a couple of different ways that endocytosis can occur. Phagocytosis is just simply having a particle that gets engulfed by a vacuole, so it kind of like a Pac-Man or something eating something else. And so then it forms a vacuole around that particle, and then that particle gets shipped off to outside the body or wherever it may need to go. So again, in this case, the plasma membrane kind of reaches out, and then eventually, once it gets all the way around that particle, it pinches off and forms a vesicle. Pinocytosis, similar. You've got these smaller particles that can gather in these little pores. They look like little pores on the cell membrane. And then when it's full or whatever is in there needs to be moved, then the vesicle just kind of slowly closes off. And once it's pinched off, then it can move away, carry off that smaller particle. There's also another version called receptor-mediated endocytosis. So there are actually these little receptors on the cell membrane. And when whatever particle molecule touches that receptor, this could be a protein molecule or a lipid or something. when it touches the cell membrane, and it basically, that receptor turns on, and so once it fills up and the receptors are all activated, then those receptors basically tell the vesicle to form. In this case, it's kind of a coated vesicle. It's got some other molecules around it. But the only difference here is there are receptors that basically tell this cell membrane when it's full to form a vesicle. And then finally, just in time for my voice to go away, is exocytosis. So endocytosis involves taking those molecules inside of the cell and transporting them somewhere they need to be inside the cell. Exocytosis means getting rid of or exiting the cell. Exo, think of exit. So these would be those vesicles that are already formed. They already got molecules in there. Could be toxins, could be proteins, whatever. These vesicles then move through the cell, and when they get to the cell membrane, then that vesicle opens up a pore. Eventually it opens up enough that whatever those molecules are can be released outside the cell. And from there, then they can go wherever they need to go. But exocytosis is taking things out of a cell. Endocytosis is taking things inside of the cell. Again, make sure you notice, right, we're talking about the cell membrane here. This is the inside, this is the outside. So endo versus exocytosis. All right, so that wraps up chapter 3. Again, highly recommend you spend some time looking at the key terms, and then even going through the chapter summary, doing the review at the chapter summary. There are some review questions that are multiple choice questions. Great review for each chapter and ultimately for your quiz. The answers for those questions are in the back of your book. And so I would highly recommend taking full advantage of those resources. As well as, again, you can easily Google exocytosis, endocytosis, tonicity, and just take advantage of what, if you're more of a visual learner, especially like I am, take advantage of using those animations and video clips to really help you out. All right, so next time we're going to be looking at how cells obtain energy.